Hello everyone, welcome to the channel Study with Asmita. Today we are going to start with the second and the final part of the chapter, the sexual reproduction in flowering plants of class 12th. So we have already covered the topic flower and pre-fertilization steps, right? So in today's video we are going to start with the topic double fertilization. Let's start with the topic double fertilization, which is a unique feature of angiosperms. As the name of the topic suggests, double fertilization. So in angiosperms, two kinds of fertilization takes place, that is the generative and the other one is vegetative. In generative fertilization, basically this is a syngamy in which the fusion of haploid gametes with haploid egg takes place, which form zygote and further it develops into an embryo, while the other vegetative is a triple fusion. The fusion of haploid polar nuclei which form diploid secondary nucleus, which is basically the fusion of another male gamete with the diploid secondary nucleus. Okay, so the formation of triploid primary endospermic nucleus, that is PEN, in the central cell takes place, and this is a nutritive tissue which provides nutrition to the developing embryo. This is known as endosperm. With the help of diagram, let us understand the process of double fertilization. So the two male gametes entering through the pollen tube in one of the synergid cell, right? And through the synergid cell, it reaches to the egg cell, and here syngamy takes place, which further develop as an embryo. Now, while the other reaches to the polar nuclei, which is already in the diploid state, and this result in triple fusion. So this is the zygote which is formed here through syngamy, and then this is the another male gamete which is reaching to the polar nuclei and form triploid nucleus, which is PEN, known as primary endosperm cell, which basically provide nutrition to the developing embryo. Now coming to the topic post-fertilization structure and events. So post-fertilization are the events after the fertilization in which endosperm formation takes place, okay, which is triploid tissue formed from triploid endosperm cell which provide nutrition to the developing embryo, and then embryogenesis, which is the formation of embryo from the zygote, which can further be classified into monocot embryo or dicot embryo. After that, seed is formed from ovule, and then fruit is formed from ovary. So a key point is here, that is endosperm is a triploid tissue formed by the process of triple fusion during double fertilization. Okay, now let's talk about the types of endosperm, which can be classified into three types. The first one is nuclear endosperm, then the cellular, and then halobial. The nuclear endosperm is formed from the free nuclear division by the only process of karyokinesis in primary endosperm cell. Example, coconut water. Then the second one is cellular endosperm, which is formed by the karyokinesis in free nuclear endosperm, which leads to the formation of cellular endosperm, that is the white kernel edible part of the coconut. Then the third one is the intermediate of the nuclear and the cellular endosperm, which is mostly reported in monocots. Now, if we talk about the endosperm, there are two cases with the endosperm, whether to get completely consumed during the embryo development or whether to remain even after embryo is developed. So, if the endosperm is remain in the seed, that is known as endospermic seed. While if the endosperm has been completely consumed by the embryo, then that seed is known as non-endospermic seed. If we talk about the example of non-endospermic seeds are pea, gram, groundnuts and beans, while castor and coconut are the examples of endospermic seeds. Coming to embryo, and the formation of embryo is known as embryogenesis, which can be defined as the process of formation of diploid embryo from diploid zygote after fertilization. Okay. Initial development of embryo are similar in both monocot and dicots, but later on some differences occur separately in monocot and dicot embryos. So with the help of diagram, let us understand the process of embryogenesis in dicot embryo. So this is the zygote here, in which mitosis division taking place for the 3 times, right? And forming the cells. The basal one is the embryo at the 4 cell stage, and another 4 are basal cell, then the suspensor cell, and then hypophysis, which are aligned on each other on embryo at 4 cell stage, right? Then further development is taking place and it forms 3 suspensor diploid cell and embryo reach at 8 cell stage. Here the peritoneal division takes place, and this is the vascular cell at the top. Then this is the suspensor cells. Then the embryo is more developed from the previous 8 celled stage. Then the further division and development forms the globular embryo. Now the development of globular embryo result in the heart-shaped embryo, which further develops and form mature dicot embryo. This is the suspensor cell, this is the plumule, this is the radicle, and these two are cotyledons. Now this is the diagram where we can see the hypocotyle, plumule and the cotyledons, radicle, and this is the root cap. Now coming to monocot embryo, development of monocot embryo is similar to dicot embryo up to the globular stage. In monocot, only single cotyledon is formed, which is at the one side of embryonal axis called scutellum. And in monocot embryo, both radicle is covered by coleorhiza and plumule is covered by coleoptile. The portion of embryonal axis above the level of attachment of scutellum is called epicotyle. So this is here the epicotyle, which is above the attachment of scutellum. Then epiblast is a small extension of embryo in monocots. Here it is the epiblast with a small extension. Coming to seed, it is the final product of the sexual reproduction in angiosperms. Seed is developed from ovule after fertilization. It is formed inside the ovarian cavity. Now the changes in ovule during the seed formation are: the first is diploid zygote, in which diploid embryo within the seed takes place, that is known as embryogenesis. Now coming to endosperm, which can be completely consumed during embryogenesis or may persist in the seed after embryogenesis. Now coming to nucellus, it may persist in some seed as thin layer called perisperm, for example in beet and black pepper. Now some changes occur in micropyle. Then coming to integuments, that are the seed coats. The first one is inner integument, that is the inner seed coat known as tegmen. Then the second one is outer integuments or the outer seed coat, that is known as testa. Coming to endosperm, if endosperm is consumed completely, then those seeds are known as non-albuminous seeds, while if it persists in some of the seed, that seeds are known as albuminous seeds such as maize and wheat. Coming to seed dormancy, it is inactive phase of embryo within the seed due to slow metabolism, or we can also define it as period from seed formation to seed germination. So after breaking the dormancy of seed, it germinates to produce a new plant under favorable conditions, right? It requires right amount of moisture, suitable temperature, and water and soil. So this is the diagram of fertilized ovule with this 3N endosperm and the zygote 2N. Now this is a diagram of a gram with a germ pore and a seed coat. This is the germinating seed with plumule, the two cotyledons and the radicle. This is a monocot seed. This is the pericarp here, areolar layer, and the endosperm here. This is the radicle. Then here is the plumule, coleoptile just above the plumule, and scutellum here, and this is the root cap. So the cotyledons here in the seed basically provide the nutrition to the embryo during the dormancy. So dicot plants have two cotyledons, whereas monocot plants have single cotyledons. Fruit is the name of mature and ripened ovary after fertilization during transformation of ovules into the seeds. Okay, now the changes in ovary during the fruit formation are: first in ovary wall, that it forms fruit wall, that is pericarp, the epicarp and the mesocarp and the endocarp. Then the ovarian cavity, it is transformation of ovules into seeds. Now types of fruits we are going to look for. So the first one is true fruit, then parthenocarpic fruits, and then false fruit. The first true fruit are developed from the ovary, such as mango. Then in parthenocarpic fruit, the fruit is developed from the ovary without fertilization. These fruit are generally seedless fruits such as banana. Now coming to the false fruit, it developed from ovary along with other floral parts such as thalamus and sepals. In case of apple, it is grown from the thalamus part of the plant, and in case of strawberry, it has grown from sepals and thalamus. A note is given here that parthenocarpy can be induced by the application of PGR, that is plant growth regulator, like auxin, example in the case of papaya and tomato. Now we are going to look at the advantages of the seed formation in angiosperms. So seeds is the basis of agriculture. It also contains baby plant inside it. It have better adaptive strategy for the dispersal so that plant species can colonize in new habitats. Dominant stage of few seeds have been formed which are very old viable seed. In the case of Lupinus articus, its dormant period is 10 years, and another example is for 2000 years is of a date palm. Now coming to the next topic, apomixis and polyembryony. Apomixis is basically a Greek word which have two parts, that is apo, which means without, and mixis means mixing, that is development of seeds without fertilization. So it is a kind of asexual reproduction which mimics the sexual reproduction in the flowering parts. So in the case of apomixis, a diploid egg mother cell is directly developed to form diploid embryo within the ovule without fertilization. Suppose this is the mother cell which directly develops into the embryo, okay, and it mimics the sexual reproduction. Now mostly it is reported in the family of Asteraceae and in some grasses. The condition where more than one embryos are formed within ovule during seed formation, for example citrus and mango. Now in such plants, some of the diploid nucleus cells develop to form multiple embryos without fertilization. Coming to importance of apomixis, apomixis seeds maintain their character over several generations, and farmers do not have to buy hybrid seeds every year. It also helps in the biotechnology to find out the gene and the characters of apomatic gene to the various hybrid varieties of the seeds. With this, we came to the end of the video and the chapter as well. If there is any doubt, you can ask me in the comment section. Till then, take care of yourself. Keep on studying, keep on enjoying. Do like, share, subscribe to the channel. Do not forget to press the bell icon. And thanks for watching.